Okay, so hi everyone. Welcome to this second appointment with the European Microglia Webinar Series. So it's really a pleasure to see many of you already connected. So as you know, this is led by two teams. I'm going just to share my screen. So the team at Achucarro, coordinated by Amanda Sierra, Federico Soria, and Marta Pereira. And on UNIL's side, we have Anne-Claire Compagnon, Fanny Martineau, Giorgio Corsi, and myself, Rosa Paulicelli. You can see here the next talks, and in case you will not have the chance to attend them in real time, you can always see them, the recorded version on this link here. Today, we have really the pleasure to host Luca Peruzzotti-Jametti, and introduction will be led by Anne-Claire Compagnon, and Fanny will be leading the Q&A. So just as a reminder, you can write your question in the chat, but also if you are allowed to ask your question directly, actually you even encouraged to do so, and you can reserve your slot by just raising the hand in the tool in the chat. So, okay, I will stop sharing my screen. Anne-Claire, to you.
Thank you. Hello, everyone. So it's my pleasure to introduce you to the speaker of today. So Luca Peruzzotti-Jametti. I hope I pronounced it well. So Luca did first a PhD in clinical neuroscience at the University of Cambridge in the UK that he completed in 2018. And later progressed to a postdoctoral position at the same university, and is now a group leader within the Department of Clinical Neuroscience and Cambridge Center for Myelin Repair since 2020. And over the last 10 years, Luca made key contribution to understanding the pathobiology of inflammation in the central nervous system diseases. And his research led to novel experimental advanced therapeutics within neural stem cells, acellular therapeutics, and small molecules for the treatment of ischemic stroke, spinal cord injury, and progressive multiple sclerosis. And Luca is now more focusing on cellular metabolism and mitochondria to find novel way to modulate chronic inflammation and favor CNS regeneration. So I think he's now giving a talk more about mitochondria and macroglia and how they maintain inflammation and cause neurotoxicity. So Luca, the floor is yours.
Perfect. Well, thank you so much for the kind introduction. Let me just share my screen. Okay. Okay, are you able to see it?
Yes. Yes.
Perfect. Okay, so I'll just start my timer. Okay, well, thank you so much for the kind introduction. As I said, it's really a pleasure to be here. So today I thought I will show you one story that we have been working on for at least 5 or 6 years. It's all unpublished up to now, but I think it's a very nice story that I think it could be important for what we're doing in our field, both in the clinics, I'm a clinician, as well as from the perspective of a basic neuroscience. So as a clinician, the patients that I see mostly are patients who are affected by multiple sclerosis. So I will just give you a brief introduction on this disease. Essentially, this is an inflammatory demyelinating disorder of the central nervous system, which is characterized by inflammation, demyelination, and most importantly, neuronal and axonal damage. The onset of this disease is in patients that normally are quite young. So they tend to have the first symptoms when they're 20, 40s, and they unfortunately have the disease for the rest of their lives. As a matter of fact, MS is the most common cause of disability in young adults. So from a clinical perspective, it's very important to distinguish what is an early relapsing-remitting MS, in which patients have episodes of neurological dysfunction, neurological symptoms, which we call relapses, which are followed by a partial or complete remission, so they go back to normal. And this characterizes usually the early part of the MS clinical course. But what happens in the majority of these patients is that despite treatments which have flourished in the past 15 years, these patients still start to accumulate a chronic level of disability over time in the absence or not of relapses, and they undergo a so-called late secondary progressive MS phase. Now, the difference between these two phases is, besides the clinical one, is related to the pathobiology of disease. So while in the early relapsing-remitting phase, the majority of the inflammatory-driven damage to the central nervous system is lymphocytic mediated, what happens in the secondary progressive MS phase is that microglia and macrophages, so myeloid cells, play a key role in this context. So these are some maps of PET with a tracer that shows how much microglia activation there is in secondary progressive MS patients. And as you can see here in the top row, compared to healthy controls, there is a widespread activation of microglia in these patients, which is by far more than what happens in the early relapsed remitting MS patients. And what we know is that this increased uptake for this radio tracer, which is partially specific to microglia, is that this correlates with neurological disability, but also with impaired cognition and cortical thinning, which are all hallmarks of progressive MS. Now, this type of diffuse microglia activation is not something that is specific for MS. We know that microglia activation is a common feature of neurodegeneration, Alzheimer's disease, Parkinson's disease, and Huntington's disease. But what has come out from recent data is that, especially in multiple sclerosis, this type of microglia activation and overall interest of the entire brain leads to an increase and accelerated brain aging, which is even more than what is seen in patients with dementia. As a matter of fact, if you compare the index between the biological age of patients and the chronological age of their brain, MS has the highest mean age gap, suggesting an accelerated brain aging. Now, what happens in MS and progressive MS, and what is driving all these phenotypes? Well, essentially, the pathobiology of MS is very complex, but what happens is that there are some type of lesions, which are called chronic active lesions, that tend to significantly accumulate over time and dominate the pathology of progressive MS patients. These were described back a few years ago, and essentially what you can see in these lesions is that there is a core which is acellular with cellular loss, loss of neurites and dendrites, and essentially loss of myelin, which is surrounded by a rim of activated microglia, which expresses markers of increased oxidative stress and reduced anti-inflammatory phenotype. Now, these first evidences were then followed by more in-depth analysis of patients. And what we could see is actually these chronic active lesions could be seen also using tools that can be used in live patients like MRI, as they are characterized by a paramagnetic rim that is surrounding this lesion. And what is now known is that these chronic active lesions, which dominate the secondary progressive MS phase, are associated with a more aggressive disease, and they are associated with the sites of chronic inflammation, failure remyelination, and axonal degeneration, overall configuring what is a picture of a smoldering brain inflammation. Now, what is smoldering inflammation? I think I would put this because it is quite important, and it's something that is becoming more and more recognized in different diseases besides MS. Essentially, it's a smoldering, meaning low-grade inflammation, which is important both in physiology, but also in response to damage. It characterizes different metabolic diseases, such as obesity, diabetes, and cardiovascular diseases, and is thought to be the initiator, but also the major driver of the progression of several disorders of the immune system, which include also progressive MS. As a matter of fact, smoldering CNS inflammation is becoming a key target for several pharma companies, which are trying to develop new therapies for progressive MS, especially because these patients, unfortunately, have limited or basically no therapeutic options, especially in the most chronic phase of disease. So what we're trying to understand in these patients is what drives smoldering CNS inflammation and how we can stop it. So as you heard in the introduction, I've been working a lot on cellular metabolism as a key driver of the activation of myeloid cells, both in physiology and in context of disease. So what happens when you stimulate any myeloid cell with a pro-inflammatory stimuli, for example, LPS, is that there are major rearrangements in their mitochondrial structure and mitochondrial function. And what is becoming more and more known is that these rearrangements prevent the repolarization of myeloid cells towards an homeostatic or an anti-inflammatory phenotype in vitro and partially in vivo, as we will see. So these are very interesting things that were being developed a lot, as I mentioned, in vitro using different approaches. And we were able to go down to the root, the biological and biochemical mechanisms that drive this transition. Essentially, during normal function, myeloid cells mostly use their TCA cycle and the forward electron transport to produce ATP. Now, what happens when you stimulate these cells, as I mentioned, with LPS or any pro-inflammatory stimuli in vitro, is that there are specific changes at the level of the TCA cycle, which include several breaks at different levels. So what happens is that these cells transition and switch from the preferential use of oxidative phosphorylation to instead the use of glycolysis. So this is very similar to what happens in cancer cells, and it's called Warburg effect. It essentially allows these cells to produce less ATP, but more fast. What happens also is that because of these breaks in the TCA cycle, there is an accumulation of succinate, which is used by the cell in different ways. One of the ways that is used is to stabilize HIF-1 alpha and induce the transcription of interleukin-1 beta, which is one of the major pro-inflammatory cytokines. The other thing that happens is that because of these rearrangements, there's an increase of the mitochondrial membrane potential, which also coupled with a reduction of the production of ATP in the mitochondria because of that switch to glycolysis, fuels a process of reverse electron transport. That means that electrons, instead of going through the normal electron transport chain, start to flow backwards from complex 2 to complex 1. And at a complex 1, they start producing ROS. And basically what happens is that these cells hijack their ability to produce ATP and transfer everything towards the production of ROS, which is mediated by complex 1 activity. Now, this is very interesting, and it's all pretty much in vitro, but how is this relevant to the in vivo settings or even to the disease? Well, to try to understand this, we adopted a fate mapping approach, which allows to differentiate microglia from infiltrating myeloid cells. And we studied these mice in the context of EIE. EIE, for those who don't know, is one of the mouse models of multiple sclerosis, and essentially similar to what happens in MS, in which you have lymphocytic infiltration at the beginning and then microglia activation more later on. You can distinguish different phases of EIE, specifically an acute phase of EIE or AAE, in which there's a lot of infiltrates, and a chronic EIE phase in which microglia are more relevant. So what we did is to use this fate mapping approach to isolate either microglia or infiltrating myeloid cells at different stages of disease and perform single-cell RNA sequencing as well as intracellular metabolomics. So basically, the data of single-cell RNA sequencing recapitulated what was already shown in 2019, but this time we added a key time point, which was not explored before, which is the chronic EIE phase, which is 50 days from immunization. And as you can see, things change significantly between control mice and acute EIE mice, where there are a lot of infiltrating cells. But then in the chronic EIE, you have clusters of microglia which return to an homeostasis phase, while other clusters tend to be present continuously, even in the chronic phases. So to try to understand what was the dynamic changes between these clusters, we did an analysis of the velocity and directionality of the RNA expression changes between different clusters. What we found was a specific cluster which was very much isolated from the other clusters, tended not to merge into other clusters, and had the lowest probability of transition into other clusters, which was suggesting of a state of persistent activation or persistence in EIE. This cluster 4 was characterized by the expression of DAM-related genes like ApoE, CTSB, CD63, but most importantly was a cluster that was high in expression of SPP1. So SPP1 was secreted phosphoprotein 1, has been studied quite well in disease-associated microglia, and recently its expression has been linked with an acquisition on an irreversible DOM state in microglia. So this was quite interesting, and that's why we try to understand a little bit more what was happening to this cluster for microglia cells. And what we understood is that the majority of the cells that made this cluster were actually present in the chronic phase of EAE. So it was a chronic EAE cluster. And then by doing subclustering analysis of this cluster, we were able to understand what were the possible mechanisms that were regulating the persistence of this cluster within the EAE mice. And interestingly enough, we found that both forward electron transport as well as reverse electron transport were among the goal terms that were most significantly associated with this cluster and these subclusters. So, as I mentioned, forward and reverse electron transport, they are can be regulated by complex 1 and complex 2. So the next thing was trying to understand what was the expression of complex 1 and complex 2 genes in this cluster. And interestingly enough, we found that while the complex 2 expression did not change in this cluster or in other clusters during EAE, the expression of complex 1 was specifically upregulated in this cluster in the acute phase EAE, and then even more in the chronic phase of EAE, which was very interesting, and it was associated also with an accumulation of SPP1 and UFS4 positive cells, and UFS4 is one of the subunits of complex 1 in the EAE mice, which was more significant in the chronic phase of EAE. So this was very interesting, but EAE is just a model of disease. So the next thing that we try to do is try to understand whether similar transcriptional profile reminiscent of this cluster was also present in single nuclear RNA sequencing data sets that were available. So we scouted the literature. We chose 2 data sets that were publicly available, the Shimmer and the Absinta data sets, and essentially looked for the expression of genes that characterize this cluster for microglia. And interestingly enough, we found that a specific subcluster of microglia in the progressive MS brain, which we call human microglia activated in progressive MS for brevity or MUMS, were indeed present in both data sets. What was interesting is that if you looked at the expression of genes that we know were associated with cluster 4, like SPP1 and complex 1, they were upregulated in this subcluster, while homeostatic genes like P2R-Y12 were significantly downregulated. And of course, we tended to look at all the genes that were present in these MUMS cluster, and essentially you can see clearly here that compared to other microglia, MUMS had a significant upregulation of SPP1, CD63, several down-regulated genes like ApoE, as well as mitochondrial complex 1 genes. So that was very interesting to see. It actually validated what we saw in the mice, but where were these cells? So thanks to the way these data sets were acquired and the sampling that was adopted, we could actually trace back where these cells reminiscent of cluster 4 were located. And interestingly enough, we found that the majority of MUMS were indeed localized at the edge of those chronic active lesions, which I've shown you before, and which are important for the type of smoldering inflammation that continues and characterizes the progression of disease. As a matter of fact, when we took several sections from patients with a secondary progressive MS and looked specifically at chronic active lesions, we could find both NGFS4 positive and SPP1 positive MHC class 2 positive cells within the lesion edge, suggesting that these cells are actually there also at pathological level. Now, this was obviously quite interesting as we had some sort of validation of what we were finding in the mouse, but still we did not understand what this upregulation of complex 1 was driving and what were the metabolic changes that were associated with this activation. So we next performed from the very same isolated cells an intracellular metabolomic analysis to try to understand how the different metabolites of these cells would change over the course of disease and among the different subset of cells. So what was striking to us was to find that essentially, while acute microglia tended to have an intracellular metabolome that was pretty much superimposable to controlled healthy microglia, in the chronic phase of the disease, chronic microglia were completely different. They were more likely infiltrating cells, and there were significant changes in their intracellular metabolome. So we then tried to understand what these changes were, trying to compare the different phases of disease. So if you compare the acute, compared with the healthy microglia, what we found is that these cells in the acute phase tended to accumulate high levels of itaconate intercellularly. The itaconate is quite important because it has been described to be a weak complex 2 inhibitor, as well as accumulation of ascorbate, the ascorbate and glutathione, which are all metabolites with non-antioxidant activities. As a matter of fact, when we looked at these sections, which we could do with the looking at the in situ metabolomics and the expression of all these metabolites, we indeed found an accumulation of all these metabolites, including itaconate within lesions in the acute phase. So this will suggest some sort of accumulation of antioxidants in the acute phase of VIE in microglia, which could act as a buffer. Instead, when you look at the chronic phase of VIE compared to the acute microglia, then things get very interesting. Essentially, chronic microglia tend to have a significant loss of ATP, much less. They have less glutathione, phosphocreatine, but they upregulate metabolites that are associated with NOS activity and reactive oxygen species production. Now, when you integrate these metabolomics data with the single cell RNA sequencing data, then you actually find interesting stuff, because besides an upregulation of F1 alpha and ApoE, which was more evident in the chronic phase of VIE, these microglia also tended to downregulate genes that are known to produce antioxidant players and enzymes. So again, this was suggesting some sort of alteration at the level of the metabolism of these cells, possibly related to mitochondrial function. So the next thing that we did was to try to understand what were the most differentially expressed genes between the different clusters. And one of the terms that came out was essentially related to positive regulation of mitophagy. So while in acute VIE microglia, there was an upregulation of genes that produce a positive regulation of mitophagy, these were significantly downregulated in chronic VIE, suggesting possibly an impaired mitophagy in these microglia in the chronic phase. So we then took this a step further, and we started looking at regulons and gene regulatory networks associated with transcription factors that regulate both the expression of complex 1 genes as well as the positive regulation of mitophagy. And as you can see there, the cluster there is all green. This was associated with the presence of regulons that regulate the transcription of complex 1 genes, but instead was completely lacking any regulon or transcription factor that was a positive regulating mitophagy, suggesting that possibly there was an impairment of mitophagy in this specific cluster and in the chronic phase of VIE. Now, obviously, when you look at changes in mitochondria, you have to understand whether there's a change in the mitochondrial content that could determine these changes. So one of the things that we did was to do a mitochondria to nuclear DNA ratio to look for mitochondrial biogenesis, and we did not see significant changes there during the course of VIE. Instead, what we found is that if you look at the mitochondrial membrane potential, this constantly increases throughout the course of VIE, peaking in the chronic phase. And this increase in mitochondrial membrane potential was associated with an increase of complex 1 activity, which makes sense based on the gene expression profile. And putting all together, we first demonstrate in vivo that what happens in chronic microglia are pathways that are all favoring what could be a mechanism of reverse electron transport with increased mitochondrial membrane potential, reduction of ATP, and increased complex 1 activity, possibly driving ROS production. So the next thing was to assess the ROS production ex vivo on these cells. And interestingly enough, chronic microglia had the highest level of ROS production, But if you gave these cells rotenone, which is a complex 1 inhibitor, you could completely block the production of ROS from these cells ex vivo, suggesting that this was a mechanism that was possibly actionable and we could interfere with to try to block the oxidative damage that is caused by pro-inflammatory microglia in vivo. So we decided to take an in vitro reductionist approach to try to understand if this was something that we could interfere with. So obviously, modeling RET in microglia is quite complicated. So we had to use different paradigms and using molecules that will basically block the production of ATP, such as oligomycin, but at the same time give a substrate for SDH so that the reverse electron transport could be favored. And essentially, we did this in different models. We used both in vitro rodent microglia models as well as human iPS-derived microglia. And the key thing is that when you stimulate and further induce RET after LPS interferon gamma stimulation, you have a significant upregulation of the mitochondrial membrane potential in these cells and the production of mitochondrial ROS. Again, if you treat these cells with rotenone, a complex 1 inhibitor, you block completely both the increase in mitochondrial membrane potential as well as the production of ROS. So that was very interesting and basically coupled well with what we saw in the EAE mice ex vivo. So we took a step forward because we could, in vitro do multiple experiments, and we decided to do some co-culture experiments using LPS interferon gamma stimulated microglia, inducing RET, and then see the effect of these cells on neurons kept in a transwell co-culture system. And that was interesting enough because basically, if you induce RET on top of LPS interferon gamma activation, these cells tend to cause significant neuronal death and reduce the neurite length of neurons that are co-cultured with them. But again, if you pre-treat these cells with rotenone, which obviously is removed upon co-culture, then you can rescue this effect. So you can block neurotoxicity in terms of cellular apoptosis as well as neurite toxicity. So this was very interesting, but again, was this really related to the secretion of ROS rather than other pro-inflammatory cytokines from these cells? Well, we did several experiments. I think the most important ones were related to the analysis of the gene expression of genes that regulate the response to ROS in neurons, such as catalase, which we found indeed being responsive to the different treatment paradigms, as well as pre-treating the neurons with microtempo, which is a ROS scavenger. And in that case, if you treat the neurons with the ROS scavenger, basically you completely abolish the effect of RET-induced microglia, suggesting that this effect is indeed mediated by ROS, and it is indeed dependent on complex 1 activity. Now, how do we move forward? Can we model RET in a better way than a very artificial, reductionist approach in which you give compounds and you can do whatever you want? Well, luckily enough, there was in 2021 a publication on Nature Communications in which these mice were described. They are called ND6 mice. They carry a point mutation in this gene. And this gene is codes for the ND6 subunit of complex 1. And essentially what happens in these mice is that forward electron transport is completely conserved, while these mice cannot do reverse electron transport. So we said, well, let's use these mice and see what happens to them. So the first thing we did is to isolate microglia, primary microglia from these mice. And here we could see already some interesting things just doing a basal seahorse experiment. Basically, if you stimulate normal microglia with LPS interferon gamma, you have a reduction of the basal respiration, which is expected. But most importantly, there's a significant reduction of ATP production, as I mentioned, one of the key drivers of RET. Well, in the ND6 mice, the reduction of ATP production was significantly less compared to wild type, suggesting that these mice were able to produce more ATP, even though were stimulated with LPS interferon gamma in vitro. The most interesting stuff is that when you took the primary microglia from wild type mice or the ND6 mice, you stimulated with LPS interferon gamma and also stimulated mechanisms like RET induction in these cells, they absolutely were refractory to RET induction. They could not upregulate the production of mitochondrial ROS, and they could not cause any neurite damage on co-culture neurons, suggesting that this was one of the killer experiments suggested that indeed RET in microglia was important for their neurotoxic effect. Now, these mice carry a point mutation, which is in every cell of the body, but still you can do EIE and compare with the wild-type mice. And interesting enough, we found that these mice tended to have a much milder EIE course. And when microglia were isolated from these mice, they displayed ex vivo a significant reduction of mitochondrial membrane potential, which again is coupled with less RET. Now, obviously, you can do several things to understand what happens in these mice. One of them is, doing single-cell RNA sequencing. So we repeated that. I think this is one of the most striking things that we found is that while in control healthy mice, there are basically no differences between wild-type and ND6 mice. In chronic EIE, two major clusters tended to completely different between the two controls. The two conditions. So cluster 1 was significantly present in ND6 mice, while the cluster 0 was present in mostly wild-type mice. And again, looking at these two clusters, one going down and the other one going up, and looking at homeostatic microglia-like markers or DAM markers, what happens in the ND6 mice is both of these classes significantly downregulated all DAM genes like SPP1, TREM2, C1qa, all genes that are very much important in pathobiology of disease. And as a matter of fact, when you looked at the number of ABA1 SPP1 positive cells expressing GP91 Tox as a marker of oxidative stress, these were significantly reduced in inflammatory lesions of these mice. So as I mentioned, this approach was, point mutation, every cell of the body ubiquitous. So we wanted to do something better and try to target complex 1 with an inducible approach, which could be first specific for myeloid cells, and second, more temporally spaced. So for this purpose, we used these NDFS flux mice, and we crossed them with the CX301 YFP CRT2 mice. And essentially, if you give tamoxifen to these mice compared to not giving tamoxifen, you basically abolish the expression of NDFS4, as well as significantly reduce the complex 1 activity, basically, I would say 80% reduction of complex 1 activity in myeloid cells. So you do EIE, you start treating these mice with tamoxifen, and boom, the EIE score of NDFS4 knockout mice significantly ameliorates over the course of the disease and remains very much lower than their controls. Again, you can do single-cell RNA sequencing, looking at how different clusters change throughout the course of disease or in health conditions. And while we could not find specific changes in healthy mice, whether wild type or knockout mice, we indeed found significant changes in clusters during the EIE. These changes were a little bit less pronounced than the ND6 mice. Obviously, the type of intervention is slightly different, but essentially there were clusters that were upregulated after NDFS4 knockout, which were essentially homeostatic clusters like cluster 1, which were also present in control mice, as well as clusters that were reduced in NDFS4 knockout mice, like clusters 2 and 6. And again, looking at gene expression of these two clusters, cluster 2 and 6, there was a significant reduction of SPP1, CD74, ApoE, TREM2, all DAM markers, with an upregulation instead of more homeostatic-like genes like CSF1R or TMN119. We also did ex vivo cytop analysis to try to understand if this transcriptional profile was actually associated with changes in the number of infiltrating cells and activation of microglia. And we found that in the knockout of the NDFS4 mice, we had significantly higher number of homeostatic microglia within the spinal cord, which is very good and would suggest that actually, interfering with this specific complex 1 subunit changes the activation of microglia and promotes the acquisition of a homeostatic phenotype. Now, we also did pathology on these mice, and essentially what we found is that even though we interfered with the complex 1, which is one of the major mitochondrial complexes. We still did not induce any death in microglia, but actually we reduced the number of 631 SPP1 positive microglia. And this was associated with a significant reduction of oxidative stress. This was calculated both on the spinal cord as a whole, as well as on ADVA1 SPP1 positive GP91 FOX positive cells. Most importantly, when you look at the effect on axons, similar to what we did in vitro, we found that both axonal loss as well as axonal degeneration was much lower in these mice, suggesting that interfering specifically with the complex 1 activity in myeloid cells after the disease onset and after the acute phase can actually rescue both the demyelination and axonal damage in these mice, which to us was very important. So we used this, several models to try to show that it was possible to interfere with RET in this pathway in microglia. But obviously, being a clinician, our goal is to try to use small molecules that could be used in the clinic to try to block this specific pathway in vivo. So essentially, if you want to block RET, you can act either on complex 2 or on complex 1. So we tested several inhibitors, which partially were produced by the group of Mike Murphy in Cambridge. Some of them were known from the literature, and another one, specifically metformin, was chosen because there are several clinical trials ongoing on MS patients using metformin to interfere with the pathology and pathobiology of disease. Metformin, for those who don't know, it has a pleiotropic effect, but also one of the effects is that it's also a complex 1 inhibitor. So the first thing that we did was to test these inhibitors in terms of cytotoxicity and effect on mitochondrial ROS production on both rodent and human-induced microglia. And essentially, we found that the single treatment with complex 2 inhibitors was quite effective. But there was another effect of using both complex 2 and complex 1 inhibition. This association yielded the most consistent results in terms of anti-ROS production on microglia. So I will not bother you too much, but essentially we did a lot of pharmacokinetic studies to try to understand which were the best inhibitors to use and how they diffused specifically into the EIE mouse brain. And essentially, while the disodium malonate was indeed accumulated in the CNS, it was also very much present in the periphery. While instead the dimethyl malonate, because of its specific chemical structure, was specifically accumulated in the CNS only and not going in the periphery. So that's why we opted for using dimethyl malonate, metformin alone as complex 2 and complex 1 inhibitors, or using the combination of the two on EAE and see their effects on the EAE score and microglial activation. So these are the data that we got. Essentially, metformin induced some sort of amelioration, which was not statistically significant in our model. DMM was partially more effective than metformin, in line with the in vitro data that I showed you on anti-ROS production, while the combination of DMM and metformin seemed to yield the most significant effect. When we looked at the ex vivo cytoph of these mice, we found something very, very interesting. So basically, the last column, the combined DMM metformin, had a double effect. On one hand, it significantly increases the number of homeostatic microglia extracted from the inflammatory lesions of these mice, and on the other hand, significantly reduced the disease-associated microglia, suggests that it's possible in vivo to induce a switch between the different states of microglia using specific mitochondrial complexes and inhibitors. Most importantly, I'm not showing these data, none of these compounds caused any significant alteration of ATP production in other organs. We tested basically 6 organs in the periphery, and there were no significant off-target effects. So that was very good. From a pathobiological perspective, and we look at pathobiology and what happens in the tissue sections, we found indeed that there was a reduction of 631 SPP1 and GFS4 positive microglia in mice treated with a combination, which was associated with a reduction of oxidative stress, reduction of axonal loss, and most importantly, a reduction in axonal degeneration. So I think I've shown you how the approach that we used in this huge manuscript, it's one single story, ranges from studying a rodent model of MS, which is the VIE and the chronic phase of VIE, and validation using human tissues. And using this approach, we could identify a cluster of specifically activated disease-associated microglia, which have high complex 1 expression in terms of genes and proteins that is present in chronic VIE in mice and is found mostly at the edge of these chronic active smoldering lesions in people with progressive MS. Mechanistically, what we show is that when microglia transition from the acute to the chronic stage of disease, they undergo a progressive alteration of the mitochondrial function, possibly related to a reduction of mitophagy, which causes a high mitochondrial membrane potential, diminished ATP synthesis, and propels RET through complex 1. We then used several in vitro and in vivo models to show you that basically exposing microglia, both rodent and human, to conditions that boost RET can indeed increase the oxidative stress and produce some sort of paracrine neurotoxicity that is dependent on complex 1 activity and is ROS-mediated. This process can be almost completely blocked by interfering with complex 1 activity using either molecules that block complex 1 or using MD6 microglia that don't have RET. And then in animal disease models in vivo, we show that interfering with the function of different subunits of complex 1, either with MD6 or MDFS4, promotes the acquisition of a protective homeostatic microglia-like phenotype, reducing instead the presence of disease-associated microglia. So overall, this work identifies a novel mechanism that sustains microglial activation in chronic inflammation, and most importantly for us, it could be therapeutically actionable. So today's news, this manuscript, which took a long time, was finally accepted for publication. And I would like to shout out to Corey and Greg, who did most of the work, and they were there from day 0 of this project. And then Christina was very instrumental for the revision process that took almost a year, but we got through to it. So I'm very thankful to all of them. And then obviously I would like to thank everybody in Cambridge and our collaborators in the UK and abroad who made this possible. And it's 40 minutes now, and I'm happy to take all your questions.
So thank you, Luca. Fantastic talk, very impressive. So Vanya,
Okay, so Emily McGuire is asking in the chat question. So she's already thanking you for your great talk. And she's asking, did you manage to check whether blocking complex 1 and 2 increases glycolysis? Also, could this explain why you don't see any difference in ATP production following blocking?
Yes, so we use complex 1 and complex 2 alternatively. Most of the time when you block one, the other one kind of like recreates the function. We did not look specifically at a glycolytic stress protocol, but we did not find significant changes in ECAR as a proxy of glycolytic function. And in terms of ATP, yes, in the wild-type mice, we show indeed that if you get primary microglia, you're treated with LPS interferon gamma, the ATP production goes down significantly. But in the case of MD6 mice, which have some sort of conserved form of electron transport and more conserved oxidative phosphorylation, then the ATP production is more sustained.
Thank you. So Agnes Najar is saying, great story, congratulations on this massive work. 2 questions. 1, do you think that RET could be the trigger of the DAM phenotype? And 2, do you think that these observations could be applied to other diseases?
These are great questions. So for the first one, I think that this paper will be opening up a chapter. We are constrained in terms of the methods that we could use to really detect RET in vivo, because these metabolites tend to be used by cells very quickly. So it's very hard to see this. My idea is that RET and FET can coexist at the same time in cells, but the preferential use of RET in chronic microglia does lead to the acquisition of a specific DAM phenotype. Now, which one comes first, we don't know. There will have to be specific experiments. We are thinking of using SPP1 CRE-RT2 mice as well to target that specific part of the microglia response. So we don't know yet, but it could be. And then the second question, could it be applied to other diseases? Yes, of course. As I mentioned briefly in the introduction, microglia activation is a feature of neurodegenerative diseases, and we are now at a beautiful time. I think in science we're trying to link what happens in a classical neuroinflammatory disease like MS, which has features of neurodegeneration on one side, and on the other side you have neurodegenerative diseases which have features of inflammation, and I think they merge together, and this could be a mechanism that can bring them together.
Great. So Doug Feinstein would like to ask if you have found changes in supercomplex formation between acute and chronic microglia.
We have not. Yeah, we have not looked at supercomplexes. We should have, and probably we're going to. We looked at mitochondrial complexes presence on Western blot, and essentially what we found is indeed most of the complexes from 3, 4, 5 tended to be downregulated in chronic EAE, while complex 1 and complex 2 were mostly present. And one of the key proteins that seemed to be upregulated was a complex 1 assembly factor, suggesting that possibly complex 1 assemblation and supercomplex as well would be affected, but we did not check that.
Okay, so I will just jump to Brian Salazar's question because apparently he raised his hand and I missed it. So maybe if you have a question and want to ask it directly, just say it so in the chat. So Brian says, amazing and comprehensive talk. I was wondering how microglia can remain so active and alive at chronic stages of inflammation like EAE, despite having a massive mitochondrial dysfunction intracellularly, so the ROS production, that does not also affect microglia themselves.
Yes, I think this is what we're trying to understand. And so how microglia acquire this chronic activation and how they can stay like that forever. So it is known that in conditions of chronic inflammation, there's some sort of clonal expansion of microglia. So as inflammation continues, microglia with I would say an advantage compared to other microglia clusters tend to stay longer. That could be one explanation, which is very much borrowed from cancer biology. And the other one is instead that there could be any epigenetic changes, even at the level of mitochondrial DNA, which has to be proven, that could cause the alteration of dysfunctional mitochondria. What we are trying to understand now is indeed if this alteration of mitophagy is something that plays a role. Microglia can stay a long term, and we've shown also in another paper that they tend to remain not only in EAE but also in spinal cord injury and acquire specific phenotypes like a foam cell-like phenotype, and possibly undergo some sort of quiescence or senescence, and that's why they stay such a long time into the EAE brain and possibly the MS brain.
So Carlos Matute says that he has 2 questions but didn't write them, so I guess he wants to ask them in person if he can connect.
Yes, congratulations, Luca, for this beautiful work and presentation. So I was wondering, you mentioned that the mitophagy was impaired in chronic EAE microglia. So correct, right?
Yes.
So I was wondering whether mitochondrial DNA leakage may play a role via the C gas stream pathway leading to interferon activation.
Yes, I think that's a key question, and I see Stefan already nodding because that's something that he's been studying in a different context in neurostem cells biology and senescence. But yes, it could definitely play a role in that.
So, and the second question was, if I understood correctly, cluster 4 microglia is key for conversion from acute to chronic EAE. So I wonder what happens to that cluster in the mutant mice.
Yes, so we looked at genes that determine that cluster, like SPP1 and other genes that, differentially express genes, and they were all downregulated. It was very hard to find a real equivalent to that cluster in the mutant mice, but all the disease-associated microglia that express genes that were specific for that cluster were all downregulated and less present. So I think the next approach would be to try to understand, as I was mentioning, with using SPP1 specific CREAR-T2 lines, whether it is possible to target that cluster as well, using the same approach that we use like in NDFS for other approaches.
Thank you very much and congratulations.
Thank you.
We also have a question from Guy Brown, who wants to ask in person.
Hi, Luca. Yeah, that was great. Great work. I was just wondering, reverse electron transport is usually invoked in a very acute situation like ischemia, where you've built up a lot of succinate, so you can reverse electron transfer to NAD to generate NADH. But you're invoking reverse electron transport in a chronic condition for days or weeks or months, which would require a continuous source of succinate and a continuous sink for the NAD. And I was just wondering how you thought that might work. Was the succinate have to come from some other cell, or how could you have a continuous source of succinate and a continuous sink for NADH in your microglia?
So it's a great question. These are all experiments that we did for, we were expecting this to be asked by the reviewers. Nobody caught on that. But we actually did these experiments. And I can tell you something. So indeed, there's an accumulation of succinate intracellularly. We did some metabolic flux experiments. It does not come from glucose or glutamine unexpectedly, but most likely comes from the aspartate, and the SDH works in reverse. We still have to understand how that works in terms of the aspartate malate shuttle, but that's actually very in line with what Chuchani has shown in the Nature paper on ischemia reperfusion. So that's confirmed. As a question regarding the chronicity of this mechanism, I think yes, in the reperfusion injuries, you have a lot of succinate build up, and then rapidly it gets oxidated and drives RET. In our condition, I think both FET and RET are present, but the predominant presence of RET in chronic microglia is the one that causes the release of mitochondrial loss.
Great, thanks very much, Luca.
Thank you.
So back to the chat, Anna Planas is saying, congratulations for this excellent talk and beautiful data. I would like to ask, how did you measure mitochondrial function of microglia obtained from the mice? Did you sort the cells? Did sorting affect microglial function?
Yes, so several ways. I think sorting the cells was the one that was the most tricky one, because we had to devise a way to still assess metabolic function, on cells that takes hours to be sorted. So I think the first 2 years of the project actually went into the fine-tuning of the protocol that we did with the Christian Pretze. And essentially, we were able to recapitulate from the ex vivo isolated microglia many of the features that we found in the in vitro stimulated microglia. So that's when we decided, yes, this approach works, and so that's when we decided to go ahead. But obviously, every ex vivo manipulation can change the metabolism of cells, and especially metabolites that can take seconds to be metabolized. So the other thing that we did in collaboration with Zoltan Tak at Imperial is to look at in situ what were the changes of the metabolism on tissue sections that were quickly snap frozen. And essentially, we found this very same changes in metabolism. So it's very important, that you kind of like understand what are the limitations of your approaches and whether you can use complementary approaches to study that. And then further going into the mitochondrial, the assessment of mitochondrial function, we did mitochondrial and nuclear DNA. That's isolation from cells, Western blot. We did also isolation of cells ex vivo, and then permeabilization and seahorse activity for complex 1 and complex 2. So we did different approaches, and I think many of them we actually fine-tuned in the PLOS Biology paper a couple of years ago. And we applied all of that thanks to the help of Mike Murphy and the mitochondrial biology unit. But it wasn't easy, but protocols are available, and if you're interested in it, I think we can talk about it.
James Woodward says, thanks for the talk. I was wondering if you started the treatment post-symptomatically. Additionally, have you looked at whether this treatment influences the morphology of microglia?
Yes. So for the ND6 mice, we cannot, because it's permutation, just confront the two lines. For the KRT2 NGFS flux mice, yes. So tamoxifen was given after 3 days after they 1 week or 3 days, I don't remember well. But anyways, after they developed the acute phase of the AE, so they started developing symptoms, they went to the acute phase, and then we waited to give treatment. And we know that at that point, after 2 days of treatment, already we can have an efficient recombination. And in that case, the curves took longer to separate because we did everything after the mice got sick. But eventually, that was clinically significant. For the small molecules, we adopted the very same approach. So we waited mice to get sick. We waited for the acute phase, which was mostly T-cell driven. And then in the transition between the acute and the chronic phase, we gave our small molecules and interacted with microglia, and that worked out quite well.
And I think we have one last question from Trevor Owens. Your treatments are designed to revert existing microglia. What about their replacement? And he means via recruitment of peripheral cells, I guess.
Yes, so that's something that we have not addressed at all, and it's something that I would like to do absolutely. Microglia exist in a different activation states, so our aim was trying to revert the activation state from detrimental to a more homeostatic state, which is something that we do in loco. But whether we can affect the recruitment of other myeloid cells or even microglia or CNS-associated macrophages and influence the disease, this is something that I don't know. And therefore, I hope to try to address this in the future.
So Aviva Tolkovsky is asking, are there differences in phagocytic capacity of microglia?
Hi, Aviva, first. And second, we did not do any phagocytic assays. We mainly, focused on, the paper was already huge. So we focused mainly on mitochondrial ROS production and gene expression, both with qRT-PCR and single-cell RNA sequencing and bulk sequencing. Essentially, what we looked at are specific genes that are associated with phagocytosis, which was actually one of the questions that one of the reviewers had at the second revision. And there were no significant changes there between the wild type and knockout. But functionally speaking, I cannot tell you, so I have no idea.
Okay, so I will take one last question from the chat, and then we will give the very last question to Rosa, who would like to ask you something.
Actually, my question was exactly this one, was related to phagocytic capacity. So question answered already.
Perfect. So Chiara Peliconde is saying, amazing talk. I was wondering if you know by which mechanism metformin inhibits complex 1. I have been using rotenone as complex 1 inhibitor to drive ROS production as positive control. So I got surprised that another complex 1 inhibitor would reduce ROS. Sorry, very interesting.
Yes, so absolutely true. Meaning that if you have, homeostatic microglia not activated, then you use rotenone or metformin, which acts on the mTOR pathway, but also on the complex 1. then you actually increase the production of ROS. So there's a nice review. I think Shallow is the first name, but essentially collects all these papers in which complex 1 inhibitors were given to myeloid cells, either in resting conditions or after stimulation. And there's a stark difference. So if you give complex 1 inhibitor to resting microglia, you tend to produce more ROS. But instead, if these cells have been pre-activated, if they're pro-inflammatory microglia, you have the opposite effect. And this is because it acts on RET. So if you were to treat your cells first with LPS interferon gamma and give complex 1 inhibitor like rotenone, then you will see the opposite effect, which is very interesting and would suggest that complex 1 possibly needs to be modulated more than completely blocked. And that's why we're looking at inhibitors that act on complex 1 transiently, because that would suggest that that's the best way to move forward. And the other thing is that you might want to use weak inhibitors, because if the complex 1 activity and dysfunction is mostly present in activated microglia, as we see, then you want the majority of the effect to be on that, and you don't want an effect on all the microglia, including the resting one.
Okay, so in place of Rosa's question, I will take one last question from that just popped up in the chat, and then we will conclude, I guess, the Q&A. So Evangelia Kyriakidou says, many congrats, great talk. I was wondering if blockade of mitochondrial ROS production blocks the cytokine production and release.
Yes, so we did this experiment. It was actually requested by one of the reviewers. We compare wild type and ND6 microglia. ND6 microglia don't have RET. Stimulate both of them with LPS interferon gamma. Cytokine production exactly the same. So pro-inflammatory cytokines exactly the same. What was changed was the mitochondrial ROS production. That's suggesting that the mechanism was very much specific for mitochondria and specific for RET complex 1 induced anti-ROS production.
Thank you, Luca. So thank you, everyone, for all of the questions. And Luca, thank you for being perfectly on time and for the very, very interesting talk. Goodbye.
My pleasure.
Thank you. And see you next month. Bye. Thank you, Luca. Great.
Bye-bye.
Bye-bye.
Thank you.